Hello, everyone, and welcome to today's webinar titled Efficient, Reliable, and Comprehensive Solution for CGT from Target Discovery to Clinical Development. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Shelley Mulock of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to genscript.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located to the right of the abstract. This survey will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Daksha Patel, Senior Segment Marketing Manager, Cell Therapy, GenScript USA Incorporated. For a complete biography on our speaker, please visit the Presenter tab from the menu at the top left of your screen. Dr. Daksha Patel, you may now begin your presentation.
Good morning, good afternoon, everyone, and welcome to my presentation. First of all, let me thank all of you for coming here today. And today I'm going to give you a brief overview about the GenScript cell and gene therapy solution. For today's presentation, the subjects can be looked under the following heading. In my presentation, I will focus on cell therapy development and gene therapy development and how GenScript can support from discovery to development journey. Let me start by sharing some background information about the company. GenScript is a biotech company that provides life science service and product worldwide. And last year, GenScript celebrated 20th anniversary. And the company was founded in 2002, and its headquarters is in New Jersey, and additional location is in China, Europe, and Japan. In 2002, launched the first custom gene synthesis service, and in 2013, GenScript launched the new subsidiary company, Best Enzyme, which is providing the synthetic biology products. In 2014, launched Legend Biotech, which is a subsidiary company specialized in cell therapy. In 2019, launched GenScript ProBio, which is a specialized CDMO service provider. In 2021, it's opened its new location and facility in Singapore and Shanghai and US. In 2022, Legend CAR T cell therapy get approval from FDA. So GenScript Life Science offer a wide range of service and product to support the life science research, drug discovery, and development. Service include the gene and peptide synthesis, custom antibody development and production, protein purification and expression, and molecular biology reagents. In recent years, GenScript launched its service in cell and gene therapy area also. For cell and gene therapy discovery and development, GenScript is providing the custom plasmid, viral vector, cell line engineering, and cell separation reagents and equipment. So after the brief introduction about the company, Next, I would like to give you some overview of the cell therapy field, history, and current status. The first observation that immune system plays a role in the anti-tumor activity is attributed by Dr. Coley in 1890. After that, it took almost a century to make the cell therapy as essential tool for cancer treatment. In 1985 to 88, tumor infiltrating lymphocyte TIL and cytotoxic T lymphocyte therapy first time confirmed their therapeutic potential. In 2002, the second generation of CAR introduced in the cell therapy field, and the creation of the CAR molecule marked a new beginning in innovation into an advancement into the T-cell therapy area. Over the last 2 decades, research on CAR T-cell therapy is progressing rapidly to improve the cancer treatment as well as to expand its use in more other cancer types and better managing the effect of CAR T-cell therapy. CAR T-cell therapy are on the verge of becoming the potential immunotherapeutic tool. More than 500 CAR T-cell therapy and 70 CAR-NK trials are being conducted worldwide, and including the 6 products which get FDA approval, and different CAR generation are shown in this figure for T-cell therapy. The first generation of CAR incorporate the CD3 derived T-cell activation. The second generation CAR carry the same activate domain, but additionally another stimulating domain added. And in third CAR generation, multiple co-stimulatory domains are incorporated into the receptor. Beside the modification of the second generation CAR, more advanced CAR immune cell therapeutics are being tested, which utilize the precise insertion of gene to avoid the graft versus host disease. In addition, CAR T cell with armor proteins expression is under development, which typically enhance the T cell function and favorably modify the tumor microenvironment. So here I represent the cell therapy workflow, and the cell therapy work process divided into the 3 area: discovery, development, commercial manufacturing. In gene and T-cell therapy, the introduction of gene could either be in vivo or ex vivo in case of CAR T-cell therapy, but most importantly, the target responsible for the genetic disease or the cancer antigen for the CAR T-cell therapy has to be identified. Subsequently, lead optimization is required to determine the affinity of cancer antigen and beta cytotoxic effect. Finally, with validation and success in the preclinical trial, many such cell and gene therapy require the GMP production. Whether you are working or developing autologous or allogeneic immunocell therapy, stem cell therapy, or gene therapy, GenScript has solution to help you to overcome the challenge you face at each stage of development. After getting the brief overview of the cell therapy, in this section of presentation, I will share some solution which is provided by GenScript to support the discovery research, including the target discovery and lead generation. Here represented the representative workflow for the target discovery and lead generation or optimization. First, the patient samples were collected, and using this sequencing technique and bioinformatic tools, potential target candidate selected for further study, and potential lead developing based on affinity ranking, and then lead can be humanized if it's needed. After CAR optimization functional assay was carried out, to confirm the toxicity and biosafety as well as the efficacy study. And as we move ahead, next I will present the GenScript product and service which can help to do this kind of research. One of them is the DNA and oligo reagents and oligosynthesis service. GenScript oligosynthesis using a semiconductor-based electrochemical oligosynthesis platform, and thousands of sequences are precisely synthesized simultaneously on cover slip size chip. Electronic control of the synthesis process ensure the top-notch sequencing accuracy, and synthesis carried out into the closed environment minimize the exposure to outside element and giving us minimum variation between the different pools. GenScript provide the DNA and RNA oligo and high throughput oligo pool and large-scale oligo pool synthesis service. And these oligo are widely applicable in molecular biology, molecular diagnosis, including the drug discovery, gene screening, and target identification. So GenScript oligosynthesis platform allow high throughput synthesis of oligo, providing a fast and economic approach to discovery of disease target. Oligo pool synthesis service offer flexible synthesis capacity, high quality oligo, and high sequence in uniformity, as well as batch-to-batch consistency. And this oligo pool can be used to construct the various library type. One of them is the CRISPR gRNA library. This fully customized, ready-to-use gRNA library help high throughput screening, including loss of function, genetic screen, or transcriptional activate gene screening. It also helps to understand the difficulties and efficiency and specificity during the CRISPR gene editing. Another one is the NGS target enrichment library, which help to detect the mutation and variant in genomic DNA occurring within the tumor and their microenvironment. It also helps to identification of T-cell receptor and assist to understand patient generic information and hence personalized medicine management. Mutant library help in protein and antibody engineering and allow the complete control of mutation ratio and create near 100% coverage of desired variant. To construct the CRISPR gRNA library, gRNA are typically cloned into lentil virus vector for different transduction into the host cell, and after cloning, library are amplified and packaged into the viruses and transduced into the host cell to generate the mutant of cell clone. And the mutant can then be screened under specific condition, such as a positive or negative, to identify important gene into the pathway. GenScript offer the variety of library for this purpose. One is a customized gRNA library, knockout library, transcriptional library, and pathway-specific library. To have successful CRISPR gRNA library, the quality of gRNA is very essential to design. And to design and optimize the gRNA, GenScript offer an online sgRNA design tool to make successful CRISPR gRNA library. High quality precision mutant library also offered for various applications. Mutant library are powerful and high throughput tools for optimizing the protein structure and function. Custom codon optimization and capabilities are also built in this platform, which allow the researcher do not only define the expression optimization for particular host organism, but incorporate specific codon and eliminate unwanted codon in their mutant library. For mutant library construction, individual mutants are generated by DNA synthesis technology, and then library are created using NGS technology. And there are 3 types of precision mutant libraries are generated: site saturation, saturation scanning, and combinational mutagenesis. And this library can be delivered as a double DNA fragment form or in the plasmid form. And this figure represents how this mutant library help to antibody engineering or screening process. For this process, the initial antibody mutant library is generated from the corresponding precision DNA mutant library by screening the mutant and affinity towards the target antigen. And bounds antibody are then eluted and recovered for their respected DNA sequence, which can be subsequently served as an initial pool of the library. And the sequence cycle can be repeated as many times as needed. Gene editing is one of the essential part in cell and gene therapy, and selecting targets are frequently validated using the gene editing to stimulate the condition to correct it. CRISPRs allow a relative way to modify manipulate gene for gain or loss function study, including the interrupt, insert, or remove. And this ready-to-use gene CRISPR kit simplify the case-mediated gene editing. It supports every steps of gene editing workflow, including the sgRNA target selection, sgRNA synthesis, evaluating the efficacy of sgRNA, gene editing, and mutation efficiency and detection. The gene CRISPR nuclease also support the RNA and DNA editing, and the Cas plasmids are also good choice for the easy use. Along with that, customized cell line also provided, and these cell lines are different type. They may be SSL line or customized SL cell line for cell power or CRISPR-Syn line. And all these cell line can support for evaluation of target, including the target gene protein identification, functional research, immunotherapy discovery, and to understand the binding affinity of antibody, protein, and immunotherapeutic target. It also supports the cytotoxicity, biological activity, and biochemical mechanism and off-target evaluation. And hundreds of cell lines delivered and used by researchers for various applications and cited in many publications. Along with that, the antibody is also a very essential research tool in scientific field and help researchers to identify and quantify their protein targets, including the protein localization, protein expression, and interaction of protein detection, as well as the cell analysis. For many applications, GenScript offer off-target antibody products, but sometimes researchers study on new diseases and mechanism, and maybe they can't find the suitable antibody for their application. And GenScript provides the customized antibody service to support the new disease and the new target research with 2 platforms. One is a B-cell cloning platform, which has a high diversity and high success rate, and Beacon single B-cell high throughput platform, which allow to do isolation, screening, and evaluation of antibody within 1 day. And this both platform are useful for generating the custom antibody for development and monitoring the CAR T cell therapy. And it's also used for customized development and therapeutic discovery. And these two platforms can make different kind of antibody, including the polyclonal antibody, monoclonal antibody, SDME generation or SCFE, and VHH generation, as well as bispecific antibody generation. Expressing the recombinant antibody in mammalian cell is essential when post-translational alternation and proper folding are required for downstream application. And to support the protein expression service, GenScript launched the Turbo CHO platform, offering the higher yield performance within the shorter period, and thus the cost-effective production and possible to create by using this Turbo CHO platform. Along with that, GenScript also providing the free codon optimization tools which facilitate the gene sequencing to obtain the functional antibody through Turbo CHO platform. High throughput E. coli platform and expression service also offered, and which is very useful for the recombinant proteins such as the cytokine, enzyme, immunogen, and antibody fragment generation. Target engagement and antibody of biomarker help to understand the relationship between the genotype and the phenotype, and SBA page and Western blotting are the most commonly used technique, yet difficult to get the persistence and obtaining the consistent quality results. For that, we're providing full range of high performance tools, including the precast gel, protein transfer system, gel staining system, automated Western blotting system, and also provide the protein and antibody purification automated system, MMAC Assay Plus, which use the magnetic bead technology to purifying the protein and antibody. And GenScript also providing the broad range of high quality primary and secondary antibody, such as an anti-epitope, immunocheck, or antibody conjugation service. And all these primary antibody are validated for different application, including the ELISA or Western blot. VHH is also one of the promising tool in the therapeutic and developing the CAR T cell therapy. And conjugation and labeling antibody also used for various applications, including the cell analysis using the FACS. Neoantigen targeted T cell therapy is a promising approach to cancer treatment, but it's still in the early stage of development. Research is ongoing to improve the identification and targeting of neoantigen and optimize the production and delivery of modified T cell. Neoantigen are unique proteins that are produced by cancer cell due to the mutation of the DNA, and to create the neoantigen targeted T cell therapy, researchers first need to identify the neoantigen expressed by the patient cancer cell. This can be done by analyzing the patient tumor DNA and RNA, and then using the computer algorithm predict which neoantigen most likely to be recognized by T cell. Once the neoantigen have been identified, T cell isolated, and then it's undergo T cell engineering process, and then modified T cells can be insert back to the patient. So here I represent the representative diagram, how the neoantigen T cell therapy works. And neoantigen peptides workflow shown next to this therapy workflow. So here the neoantigen peptide is produced into the GenScript shows here, and peptide can be used for various applications, including the immunogenicity testing and screening, clinical stage vaccine development, and post-clinical efficacy study also. And GenScript has providing reliable peptide from last 18 years with our pro-priority microwave-assisted peptide synthesis platform, now able to deliver the high-quality peptide within the 5 days. custom peptide synthesis service also available, and we are providing different kind of peptides quality, fast peptide, which are suitable for targets identification, and basic peptide, which are ideal for immune monitoring and T-cell epitope mapping, and premium peptide service also available. We also offer the free online peptide library design tools, which help to generate the different peptide library based on your application. And peptide library allowing scientists to identify the most important peptide for binding interaction quickly. For example, in case of T-cell application, for cancer, libraries have been used to identify specific ligand which can recognize the tumor cell. And to meet the different purity demand for multiple application, we developed the two different type of peptide library. One is a standard peptide library, which is available in high purity, and the microscale peptide library, which is ideal for preliminary screening and application. Overall, GenScript is offering wide range of products and service for target discovery and target identification. Genetic association, protein expression, functional screening, and genetic manipulation of target can be identified or can be discovered by using all these different tools which GenScript is providing. In this section, we'll share more detailed information about the GenScript solution for cell isolation, activation, and expansion. So cell therapy are an emerging treatment option for wide variety of disease. And most cell therapy involve the cell isolation in laboratory setting. For cell therapy, drug discovery, research, and in core facility as the part of the cell manufacturing as well as cell banking workflow, the cell isolation reagents are essential. Furthermore, manufacturing high quality, consistent, and efficient cell therapy product depend on obtaining high quality reagents from established manufacturing partner with supply chain security. GenScript has developed the cytosine reagent and consumable, including automated platform system, which gives the high purity, high recovery, and functional viable cell to support the cell therapy workflow. So here I present the cytosine 1000 equipment, which is a cell isolation equipment and work on basis of immunomagnetic cell isolation concept. It's automated instruments, and it has the key highlight features which include easily accessible, barcode scanning for reagent entry, customizable programming, fully traceable. Traceability means we can trace the whole process and from during the manufacturing workflow, as well as it's allow the workflow exact situation, means workflow screen provide real-time monitoring, and it's walk-away solution. And it is providing two different type of tubing set. One is from the isolation, another one for the depletion. And within the isolation, there is a two different type of tubing set, which giving you allow to purify two different number of cells, starting from 6 × 10⁻10 versus 1 × 10⁻10. And cytosine platform and reagents enable researchers to scale up their operation and can be easily integrated with other instrument and platform for all necessary processing steps, including the sample washing, labeling, magnetic separation, and cell concentration. So this is representative workflow, and it's well suited for the modular system. And these reagents are efficient, sterilized, non-toxic, and easy to use. Here represented one representative data for using the cytosine or 1000 equipment, where cells are isolated using cytosine equipment along with the control experiments carried out with the another instrument which is available in market. And if we see here, the data are shown into this slides, where cell viability are not effect after the cell sorting, and the purity and recovery are comparable for CD4 and CD8 both cell and working. So cytosine 1000 equipment working very well in terms of purity, recovery, and viability. And cytosine manual separation systems allowed flexible and convenient option for large to small scale separation. So this one used for the research work purpose. And we are providing different cell isolation ready to use reagent. They are available in REO grade as well as GMP grade, including CD4, CD8, CD3, and NK cell isolation, as well as TCR alpha beta nanobeads. We also providing the streptavidin beads along with the biotinylated antibody and T cell activation reagents. So here we presented some comparison data for pre and post isolation from leukapheresis using GenScript reagents, and we used another competitor reagents as a control, and purity and recovery of CD4 and CD3 in both the cases looks quite promising, including their performance is matching with the control samples. And this reagent can also be used for the depletion purpose. And here we presented CD4 depletion using the human CD4 cytosine nanobead, and it depleted CD4 cell by 99.9%. And it's showing very similar data as the control sample. And now I'm talking about the T cell activation reagents. So GenScript T cell activation reagents is uniform, biocompatible, and non-toxic CD28, CD23 couple antibody couple nanobeads, which allow the feeder cell-free and antigen-free activation and expansion of T cell from PBMC or enriched T cell population. And this is ready-to-use reagents are very handy. They are small size and appropriate to use for scalable application for clinical manufacturing. So activation reagent study represented here. In this experiment, CD4, CD8 cells are isolated simultaneously and then use them for activation purpose. And the figure A represents the expression of the activation marker after the 48 hours, and cell stained with the fluorescence dye after the 7 days. It shows the proliferation is happening. So compared to control, if you see there is a shift into the FACS plot. And then this is the T cell expansion fold data, which is represented in figure C. And we can see the activation, NCT cell activation reagents are data comparable with the control sample. And along with that, flow cytometry analysis of T cell subpopulation also shown. And they are also, overall data looks quite promising. In this experiment, the T cell activation by using cytosine CD3 nanobead purified T cell is used as a starting material. And the figure A represents the expression of activation marker. And then figure B represents the higher fold expansion is observed. And then figure D represents the T cell subpopulation. So same trend we can see as the previous experiments by using the NCT cell activation reagent. And this is confirming that NCT T cell activation reagents giving very good results for the activation, and then expansion is also very good. NK cell enrichment carried out by two methods. One is CD56, direct selection without any kind of depletion, and the purity and recovery represented here, and the control sample and the cytosine CD56 beads performance are very well. And then NK cell enrichment also carried out after the CD3 depletion. So overall, all the reagents performing very well in different application and different platform. And this GenScript TCR alpha beta beads. These beads support the one-stop isolation process. So via direct labeling, need only one reagent, which is antibody coupled particle available. So anti-TCR alpha beta antibody couple on the particle surface. And the advantage is like easy to use in the bigger scale, means auto scale, as well as it's a simple one-step process. While other reagents which is available in the market, they are indirect labeling. So first you need to label the cell with the antibiotin antibody, and then wash the excess of antibody, and then add the biotin label antibody and SAB particles required for magnetic isolation. And these are the preliminary data. So TCR alpha beta bead directly labeling is also performing very well. And this bead has many advantage because it's only using single labeling. So cytosine and growth factor also one of the essential reagents, and in T-cell therapy required the growth factor to expand, which are provided by the using the media or by the serum. And one of the useful growth factor is IL-2, which is binding in receptor and promote the T-cell expansion. So GenScripts offer the comprehensive catalog, recombinant protein, and cytokines, and with the lot-to-lot consistency. And this can be used for various applications, like for the immune cell culturing, stem cell culturing, and even they can be used in the cytokine therapy. So more than 2,000 cytokines, growth factor, and peptides for cell therapy research are also available, and we also providing to the custom synthesis service. After getting information about the cell isolation activation, in this section, I will give more detailed information about the cell engineering and gene therapy platform, including the viral gene therapy and non-viral gene therapy platforms, which GenScript is providing. So before presenting the GenScript cell engineering product and service, I briefly introduced the T-cell engineering progress. And in CAR T-cell therapy, T-cell engineering is the essential to obtain the efficient tumor killing property. And originally, the viral vector have been used for CAR T-cell manufacturing. However, the random gene integration introduced immunogenicity risk associated with it. And later on, CRISPR T-cell CRISPR T-cell use along with the adenovirus vector to deliver the DNA payload for target insertion via HDR template into TCR. But despite of its good performance and high editing efficiency, limited vector capacity and high manufacturing cost, it's restrict the viral engineering workflow. And currently, non-viral CRISPR editing providing an alternative tools for gene insertion. And the first CRISPR-based therapy phase clinical study is also complete. And to support this, the development of CRISPR genome editing opened the new possibility in precision medicine. Currently, there are number of research is under progress, including the blood disorder, cancer, diabetes, infection disease, and protein binding disorder. and number of CRISPR clinical trial is under progress, and it's going very fast day by day. So to support ongoing demand for non-viral system, GenScript offered a wide range of reagents to support the discovery work. And we already discussed about all these reagents in the discovery stage portfolio. But along with that, GenScript offered the RNP, including the synthesis sgRNA and Cas9 protein, and the DNA payload and donor templates, including the ssDNA, stable dsDNA, and GenCircle dsDNA. CRISPR-based gene editing application moving from basic research to clinical manufacturing, high-quality raw materials, and combined along with the complete documentation are becoming more essential to fulfill high-quality specification. GMP grade GenCRISPR Ultra Cas9 service also offered. We offer 2 type Cas9, and key highlights of this reagent: it's providing high editing efficiency, high lot-to-lot consistency, ultra high stability, and high quality specification. In this figure, it shows that T-cell receptor alpha-beta chain knockout efficiency was measured and analyzed by the FACS using the GMP grade Cas9 system, and off-target efficiency also measured into 2 different platforms, OT1 and OT2, and GenCRISPR Cas9 shows low off-target efficiency. We also providing 2 sgRNA service line. The first one is the Easy Edit sgRNA, and this sgRNA is excellent option for CRISPR screen, and significant benefit of service are highlighted here because it's ready to use and ensures high knockout efficiency. Another one is a Safe Edit sgRNA, and these are the HPLC purified. And so this service line is ideal for gene therapy application as well as using CRISPR in animal model. And the main benefit of our safe gene editing is high purity sgRNA with modification ensuring the minimum cytotoxicity and off-target for primary cell and stem cell. So as mentioned in the previous slide, the main benefit of our safe edit sgRNA include high purity. So here is represented like it's showing 99% high purity compared to the commercially available other sgRNA, and HPLC purification remove the presence of unwanted or truncated sgRNA, and which can reduce the off-target effect. So purity of GenScript shown here is 99%, which is very high compared to others reagent. Another powerful application of CRISPR-Cas9 genome editing technology involved the DNA sequence via HDR pathway, and many factors may affect the efficacy of this approach, and choosing of the suitable HDR template is the essential. And GenScript offer the high quality sequence verified HDR template, including the gene extract ssDNA, GenVend dsDNA, and GenCircle dsDNA. They all are available into RUO and GMP-like, and each of them have a different insertion length capacity. With advancement in the CRISPR-Cas9 technology, it is now possible to insert the target gene sequence at the specific genomic site in human primary T cells for CAR expression without using the viral vector. So in this case study, Dr. Brian developed a hybrid single standard DNA, ssDNA, HDRT with CTS. And the diagram of this hybrid ssDNA HDRT shown here. And the study was conducted at the Markinson lab at UCSF. Due to the challenge in the production of the long ssDNA sequence in the lab, Dr. Bryan collaborated with GenScript and were able to perform therapeutic gene editing on primary T cell at clinical stage using GenScript ssDNA. And the results shown, so in this slides, I present the few data of that experiments. So figure A represents the diagram of non-viral CAR T cell manufacturing process they use, and the result shows that GenScript SSCTC consistence performs better compared to the lab-generated SSCTC and ssDNA, which is represented in figure B, C, and D. And the knockout efficiency in GenScript SSCTC via electroporation without enhancer can reach up to 46% at clinical scale, means on this scale. And the knock-in positive live cell count generated by the GenScript SSCT was above estimated patient dose, and in vitro assay demonstrate the efficiency of killing BCMA CAR cell is shown in this graph G. So hence, the Dr. Bryan from Marson's lab developed the novel payload for non-viral approach to gene editing, and it's applicable in clinical scale to support the GMP compatible non-viral CAR T cell manufacturing using GenScript ssDNA. GenCircle ssDNA is also a novel small circular double-stranded DNA vector, and the GenCircle ssDNA include its advantage is like it's safer because its antibiotic resistance gene and bacterial origin sequence has been removed. It's a smaller and simple, so smaller vector backbone generate the low cytotoxicity and immunogenicity and offer better stability. And it can be used in application like GenCircle dsDNA as a knock-in template, as well as viral packaging plasmid and GOI non-viral vector. Overall, more efficient GenCircle dsDNA can be offered from GenScript in two different quality, and this can be very useful for gene and cell therapy study. Here we have data comparing the GenCircle dsDNA vector compared to the traditional plasmid, and as we can see, HDR knock-in efficiency is significantly higher with GenCircle template. It's about 30% improved compared to the standard plasmid, and low cytotoxicity is also observed, and hence this performing very well, where means gene editing can be advanced into cell and gene therapy using GenCircle dsDNA. mRNA is also now becoming new therapeutic approach for a range of disease and simplifies cell-free manufacturing, cell cycle independent performance, and risk-free insertion of the mutagenesis increase the IVT mRNA adaptability into the therapeutic field. GenScript solution to mRNA production start from the gene synthesis to IVT mRNA production as shown here. And to simplify, to simple and innovative mRNA manufacturing workflow, offer more opportunity for personalized medicine use. And mRNA produce are optimized with the proprietary platform, ensuring the mRNA quality and expression efficiency. Custom mRNA service also available. Custom mRNA service also available, and they are 3 different type of mRNA we are providing. And this here represented the mRNA mediated delivery of Cas9 has certain advantage. It's efficient and there is no genomic interference happening, and it's providing high editing efficiency and potential low off-target rate. So here shows the preliminary data where Cas9 mRNA used for the same purpose. And as shown in the figure, mRNA integrity impact its expression. And so high integrity support the high expression for Cas9 mRNA system. And this version of Cas9 nuclease reduced the off-target effect by over 10-fold while maintaining the robust on-target genome editing efficiency. So mRNA products including gene editing mRNA, Sleeping Beauty mRNA, and reporter gene mRNA also available. So as a leading cell therapy nucleic acid provider, we believe in support the future of non-viral gene therapy and providing all different grade reagents for discovery to clinical application. And their different quality are mentioned, like for R&D grade, process development, and clinical manufacturing grade available for non-viral gene editing as well as gene therapy purpose. We also providing the third generation lentil virus packaging to ensure the downstream safety for CGT application. Difference between the two generations shown in this table. So the full spectrum of service available from for LLV, and so that's including like optimization of viral production to obtain the maximum yield, and intact functional viral vector with high transduction efficiency, and multiple titer and QC methods to confirm the consistent quality. And lentil virus ELISA kits also designed to measure the physical titer, and it makes lentil viral titration easy, accurate, and reproducible. And this kit can be very useful tools for routine quality control and lot-to-lot release and purification purpose optimization. Along with the lentil virus packaging service, we're also providing the AAV packaging service, and all these can be used for the knockout screen, cell engineering, and therapeutic gene delivery. GenScript ProBio is a CDMO service. It's providing the CDMO service for CGT application and all the customized option available. And the most common used material in gene therapy development and cell therapy development is plasmid. So it's also the GenScript ProBio providing the service for plasmid of any size for various applications. So it's including the GMP plasmid production in different quality, and also like service accommodate both the small scale as well as the large scale requirement. Similarly, lentil viral vector is also a useful vehicle to deliver the gene and into the cell and gene therapy area. And the safety and stable manufacturing of LLV is very essential. So GenScript GenScript ProBio support the developing both the adherent and suspension platform to delivering and manufacturing the lentil viral vector. AAV vector is also most widely used as a vehicle in gene of insertion in vivo gene therapy. And AAV service cover the various needs from research to IND application to clinical trial. So ProBio division is providing all this service, customized service, and also providing the CAR lead discovery service and CAR-T in vitro assay study. And along with the plasmid manufacturing service, we last year launched the automatic large-scale high-throughput plasmid purification instrument, and along with their consumable, and the MLAC Quattro is a fully automated solution for purifying the large-scale, high-quality plasmid. And the comprehensive solution consists a system controller that provides the simultaneously operation of up to 4 independent purification module. The M Quattro powered by magnetic bead technology, and the key benefit is allowed the high throughput up to 24 sample and flexible volume ranging from 50 to 200 ml and ready to use consumable available for that. So GenScript Biotech, premium, convenient, and reliable service and product used by number of researchers and scientists worldwide, and more than 74,000 peer-reviewed journal articles have been cited GenScript Biotech service and product. So this brings me to end my presentation, and in summarize, my main points covered is like we offering end-to-end solution to accelerate journey from discovery to clinical research and commercial manufacturing by providing the high quality service, reagent and instruments with scientific and regulatory support. Here I show some key reagents which use both in gene and cell therapy, and their different quality available for different stage from target discovery to lead generation and up to preclinical to clinical stage. So GenScript offer the customized option for many products and service, and along with that, in these slides, I just to extension the service specifically for cell therapy field, which include the cell isolation, activation reagents, and we also providing the protein purification and plasmid purification system. So thank you all for your time and interest, and I hope that you find this useful information. If you have any further question or feedback, please don't hesitate to reach out us. And I will now able to answer any question if you have. Thank you.
Thank you, Dr. Daksha Patel, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. So, Dr. Patel, the first question is, what tools for CAR optimization do you offer?
That's a good question. So, for CAR T-cell therapy, in which generally the T-cell engineering is very essential, and it's required to make sure the off-target effect is reduced and enhancing the efficacy. We providing like precision mutant library, and which help to make the random mutagenesis less random, and it's also allowed to control the DNA synthesis with semiconductor technology. And this technology will give like its company's proprietary algorithm design tool can help researcher to select the best antigen binding domain and to co-stimulatory domain for the CAR T cell, as well as along with the precision mutant library, GenSmart codon optimization toolkit is also a free online tools which allow to design the best codon for the CAR T cell efficacy and targeting efficiency improvement. Along with that, DNA construction design tools, that's also a very useful to make the library module for designing the DNA library and then create the new multimodal multi-domain protein or optimize the protein function or generate the recombinant protein or antigen. And CRISPR gRNA library also very useful to screen maximize their efficiency of the CAR, which is designed on the cell surface. as well as the gene editing technology may also help, like CRISPR-Cas9 gene editing tools, and can enhance by CAR-T efficacy. So overall, we are providing like number of customized solution, including like some reagents as well as services to design CAR-T and optimize the efficiency and specificity of CAR-T cell.
Okay, wonderful. Thank you. The next question is, do you see any toxicity effect of the T cell activation reagent?
Yeah, that's a good question. So generally, it is necessary to understand the toxicity effect of T cell activation reagent. But the T cell activation reagent toxicity also effect on the depend on the concentration, duration of exposure, and the type of how long, means with which cell is your starting material. But in our company, before launching this reagent, we did systematic study, including we doing like regular basis of physiochemical characterization of this reagent. And in vitro assay was also carried out to understand that T cell activation reagent has no effect on cell viability as well as proliferation or cytokine production and other relevant parameter by doing certain type of assay. And we also did in vivo assay using the third-party service, which include the safety, efficacy, and pharmacokinetic study, and which to meet the regulatory compliance. And if you're looking for any kind of these documents for any stage, we definitely happy to provide that all the safety data, which is we created in our company during the launch and after the launch of this reagent.
Oh, that's very helpful. Thank you. Okay, so our next question is, how long is the delivery time for the LLV packaging service, and what type of QA are you providing?
So for LLV packaging service, we have like 3 different type of packaging service. One is a regular titer, and so and the another one is a high titer, and one is a ultra high. So basic QC method we providing is ELISA and microplasma testing, but we are providing additional QC testing also, like sequencing and FACS analysis, and it taking mostly 2 to 4 weeks for all this service to deliver. And definitely, if you are looking for this kind of service, feel free to reach out. We will make sure you get the reagents within your time period.
Excellent. Okay, well, it looks like we have time for one more question. So the last question is, are you able to provide demo for CytoSync 1000 equipment?
Oh, yes, we are happy to provide the demo. And we also giving the reagents initial testing if you want to do in your lab as a beta tester, so you can test them. And once you like, then we can come and provide the CytoSync equipment demo along with the reagents. So definitely we love to provide you demo of the equipment.
Perfect. Well, thank you, Dr. Daksha Patel. Do you have any final comments for our audience?
So thank you, everyone. And please feel free to reach out. GenScript is always there to support you.
Wonderful. Thank you again, Dr. Daksha Patel, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors for sponsoring today's webinar, GenScript being our sponsor. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.